Hello there. So I first want to thank you for taking the time to check into my presentation today. My name is Kyle Helms. I'm a Mark Research Fellow in the Department of Biology. I work in Dr. Jennifer Curtis's Cellular and Developmental Biology Lab. And so today I'm going to be talking to you guys about the interaction between the transcription factor T-shirt and the transcriptional co-regulator CtBP during Drosophila melanogaster eye development. So here's my abstract. This is kind of just a big picture, including some background and kind of what's going on with this project today. So you guys can go ahead and take the time to pause the video to read through it, or we can go ahead and get started. So before I jump into what exactly I've been doing here, I wanted to give you all some background into the project itself. So as I mentioned earlier, I'm using Drosophila melanogaster, or fruit flies. And fruit flies are an important model organism in studying development because they're very genetically similar to humans, and that a lot of their genes are conserved across into mammals, including humans. And there's a wide variety of genetic and biochemical tools that we can utilize to study development in Drosophila. And so we know that the regulation of gene transcription plays large roles in development, specifically in tissue specification and tissue-specific proliferation. Now, the way in which these cells are able to control gene transcription is through sequence-specific transcription factors like ILIS and T-shirt, which we're going to look at today. And so both of these are required in eye specification and proliferation, but they're only expressed in proliferating eye precursor cells. So if we take a look at the right, we can see a diagram of the developing Drosophila eye, and we see demarcated anterior, MF, and posterior. And so MF is the morphogenetic furrow, or a physical indentation in the eye disc itself where cells that are proliferating are transitioning into differentiated cells. And so we see that in this area of proliferation, we see that expression of ILIS and T-shirt. However, in between them, we see the expression of CtBP, that C-terminal binding protein, which functions as their transcriptional co-regulator, and it works to attach transcriptional machinery to these sequence-specific transcription factors in order to regulate transcription during development. But we see that here it's expressed throughout the entire eye disc. So the objective of this project, or rather the question we're trying to answer here, is whether or not T-shirt and CtBP are interacting during fly eye development. And so we sought out to answer this by looking at physical evidence, including glutathione S-transferase pull-downs and co-immunoprecipitations, and also looking at genetic evidence, so epistasis, so gene expression, and those co-immunoprecipitations again. And so the next kind of question in this series was, are there other proteins that are interacting with this T-shirt CtBP complex during development? And so future co-immunoprecipitation and mass spectrometry experiments will hopefully answer this question. So the first of the methods that we used to answer these questions was genetic epistasis. And so essentially this is utilizing the GAL4 UAS system to drive the overexpression of the CtBP gene in these fruit flies. And the way in which this works is this GAL4 is attached to a tissue-specific enhancer, so in this case an enhancer specific to the eye. And so this drives the expression of the GAL4, which will then bind to the UAS, which is the upstream activating sequence of the GAL4 system. And so once these are activated, it'll drive the expression of CtBP. And so we'll get this overexpression of the CtBP gene in these fruit flies that have this GAL4 UAS system functioning. So I also used GST pull-down assays to show that there is a direct physical binding between T-shirt and CtBP, at least in vitro. And so the way in which these GST pull-downs work is there is a prey plasmid and then a bait plasmid. So in this prey plasmid we have this vector that is holding the cDNA of T-shirt, and then in this bait we have this vector that's holding the CtBP cDNA as well as GST, so this glutathione S-transferase segment inside of this plasmid. And so once they're induced by isopropyl thiogalactopyranoside, we should see that there is an interaction. And if they're bound after washing and then eluting, we should still be able to see this complex of this GST and then our bait protein and our prey protein bound to the beads when we precipitate them. And so we did this both ways, using t-shirt as the prey and then t-shirt as the bait, and vice versa. So we're also able to tag the t-shirt gene itself with the green fluorescent protein tag. And so we did this by using the CRISPR-Cas9 system. So essentially this CRISPR portion of this Cas9 system was able to guide these sort of molecular scissors, the Cas9, to the point in which we wanted to insert it into this genome of the t-shirt gene. And so we can see here in this diagram that we were able to insert this EGFP tag directly in front of this t-shirt gene. And so when we do experiments like co-immunoprecipitations or other biochemical assays, we should be able to use antibodies against this EGFP and be able to see this complex when experimenting. So in order to show that there was a physical binding between t-shirt and CtBP in vivo, I used co-immunoprecipitations. And so these start with a cell lysate. And so my lysate consisted of about 150 Drosophila eye discs. And so yeah, I did have to go in there and dissect these larvae and remove both their eye discs and get them in this lysate. And so the next step is incubating this lysate in a separate Eppendorf tube that already has antibody and G protein beads. And so this G protein is just a generic protein used in this co-immunoprecipitation. And so you let it incubate with these antibodies and beads, and in this case the antibody is an anti-GFP. And so once they're bound or they're allowed time to incubate, we then wash it, and what you hope to bring out of this is this complex between the G protein beads, the antibody, and then the complex bound to that antibody, the protein complex of interest. And so we kind of go ahead and see if this functioned correctly and see if we did pull out the t-shirt and CtBP complex by Western blotting, because we'll go ahead and see bands that are consistent with protein levels of CtBP and t-shirt. So now that we've walked through the methods, we can go ahead and jump into the results. And so this first slide of results is the results from the genetic epistasis experiments. And so we can see here that the first part of these experiments was the overexpression of t-shirt. And so we can see that in flies where we overexpressed t-shirt, there was significant tissue undergrowth. And so we can see in A that there is actually no eye phenotype in these overexpressed t-shirt flies. In B, we can see that there's a one small eye phenotype. And in C, we can see there's a 2-eye phenotype on the same side of the head, and both are significantly small. The next part of this was these overexpressed t-shirt flies with the depletion of CtBP. And so when we depleted CtBP, we did see similar results, and that there was either no eye, 1 eye, or 2 eyes. However, when we take a look at the left to this graph, we see that the y-axis is the number of flies that were used, to our n, and then the x-axis is showing us which group we're looking at. So this first bar is this overexpressed t-shirt alone. And so we use this doing a chi-squared test. And so in red, we can see the number of 2 eyes, and green is the number of 1 eye, and blue is the no eye. So we see that there's a pretty significant number, almost around 20, flies that have no eye. However, when we take a look at the eyeless t-shirt CtBP flies, so these overexpressed t-shirt and depleted CtBP, we see that that number of no eyes is actually decreased significantly, so much so that the p-value is actually equal to 0.0005. So this just shows that this difference is statistically significantly different and lower than the t-shirt overexpressed flies alone. And we can also see that the 2I phenotype actually grows as well. So we see almost this adverse relationship. Now in this slide, we're going to go ahead and look at the results from the GST pull-down assay, where we're able to show that there is a physical binding between t-shirt and CtBP in vitro. And so we're actually only going to take a look at C and D because A and B are a different part of the project that isn't being presented today. And so when we take a look at C and D, we can see in column 1, this is just a load. So this is in vitro translated products. And so this is where we should see that fused protein, either for t-shirt or CtBP, in the row. Now 2 is just the GST protein mixed with prey protein, so that functions as our control, because since it is the prey, it's not bound to that GST, and so we shouldn't expect anything in that second column. However, in the third, this should be that complex of our pulled down prey protein with the GST fused bait protein. So when we see that when we're using antibodies against t-shirt, we're able to identify that using GST CtBP as our bait, we're able to pull down a complex of t-shirt and CtBP. And then in D, we can see using GST as our bait, we can see that there's a CtBP marked in the Western blot. So this is showing us that in vitro, we were able to recover some kind of physical complex or interaction between t-shirt and CtBP. Here we're looking at the Western blot analysis of the co-immunoprecipitations that we used to see if there was a physical binding between t-shirt and CtBP in vivo. And so this Western blot specifically is identifying whether or not we were actually able to even pull down this t-shirt GFP complex. And so that GFP is from those CRISPR-Cas9 constructs that we created earlier and that I talked about earlier in the presentation. And so this Western blot is from a lysate that consisted of about 75 ion channel discs. And so we can see that in column 1, this is just crude lysate, so this has not gone through a co-IP or has any beads present, and so we don't see anything there. If we look at column 2, these are just the protein beads alone, and so this works just as another control, and we don't see anything in that column as well. However, in column 3, this is the composition of our co-immunoprecipitation product, our lysate and our protein G beads, marked with an anti-EGFP antibody. And so we can see here that we have a band at around 75 kilodaltons. And so we would expect that GFP in combination with t-shirt would have a band around 130 kilodaltons, just because both protein sizes put together, if they're in complex, we would see them higher theoretically. However, other studies and previous Western blots in our lab have identified that we really see t-shirt closer to around 75 because the protein itself is very fragile. So now we're going to go ahead and take a look at the Western blot analyses of the co-immunoprecipitations that showed the physical interaction in vivo of t-shirt with CtBP. However, this Western blot is using an anti-CtBP antibody instead of an anti-GFP. And so we can see in this first column that this is just the crude lysate of the antennal discs. And then in the second one, we can see that this is the collection of the co-immunoprecipitation, the protein G beads, and the antibody. So the antibody used in the co-immunoprecipitation was an anti-EGFP. And so we use that one in the co-immunoprecipitation, but the anti-CtBP antibody in the Western blot. And so this is in hopes that when the protein was combined to the protein G bead and the antibody, it was combining via that complex of the EGFP t-shirt. But now that we're using an antibody against CtBP in this Western blot, we would hope to see a band marking for CtBP, which would insinuate that there is a complex that is pulling CtBP out of this co-immunoprecipitation. And so in that third column, this is our negative control, and so this is just the crude lysate and protein G beads. no anti-EGFP antibody. So we can see in the second column that we do have a band that's visible around 50 kilodaltons, and that's what's expected of CtBP. So we can see it there around 50 kilodaltons. So this is just further showing us that there is an interaction between CtBP and t-shirt, and that when we're co-immunoprecipitating them and we're pulling down this complex between EGFP t-shirt and CtBP, and then by using this Western blot, we're able to identify that they are interacting in some way. So we can see on this slide that this is just a continuation of this Western blot analysis of the CtBP t-shirt interaction in vivo. And so essentially what this one is showing us that we were able to replicate the previous Western blot analyses, and that we were able to show that there is a band that comes down again for CtBP when we are using an anti-CtBP antibody after a co-immunoprecipitation using this anti-EGFP antibody. The band here is not as strong, however, it is good evidence that we are able to replicate the experiment. Now, what we can gather from the data that I've presented to you all today is that there is most definitely an interaction during eye development between the transcription factor t-shirt and the transcriptional co-regulator CtBP. And so this physical and genetic interaction has been evidenced by the genetic epistasis, co-immunoprecipitations in Western blots, and GST pull-down assays that I performed. And so some future aims in my lab is to use mass spectrometry to further offer insight into what other proteins are working with t-shirt and CtBP in complex to regulate transcription and therefore development. I'd like to take the time to acknowledge that my research is supported by the Maximizing Access to Research Careers MARC program here at NMSU and the National Institutes of Health under the award number GM07667-41. I'd also like to thank Michael Burnett for helping me in the lab and really directing me with the experimental protocol involved in a lot of these experiments. So a big thanks to him. I'd also like to thank the Bloomington Drosophila Stock Center for, of course, providing the fly strains that we used in this project, and the David Arnosti Lab for gifting us with the anti-CtBP antibody.